You have been asked to produce a semen sample for a scientific study. Now, in order to collect a good, healthy sample that can be compared to others in the study, you will need to know some basics on when and how to collect the sample. Don't have any kind of sex or masturbate for 2 days before you collect the sample. The number of days without sex will greatly affect the number of sperm within your sample. To collect the sample, masturbate directly into the glass jar. Don't use a condom, rubber, or any other container. Don't use lubricants such as baby oil or other lotions, and don't collect the sample by withdrawal during sexual intercourse. The amount of semen ejaculated is small compared to the large jar. This jar is used because its size makes collection easier. Don't be concerned about how much there is in the jar. After you collect the semen sample, close the jar, fill out the information on the label, date and time, whether any missed the jar or was spilled. A few drops after the main ejaculation may miss the jar, and that's not important. It is important if a large portion is lost. Also, write down the number of days since you last had sex or masturbated. Your identification number is attached to the jar. When you filled out the label, put the jar into the thermos container and close it tightly. The thermos is provided to protect the semen from temperature changes. The thermos and jar should be kept at room temperatures both before and after the sample is collected. Bring the thermos to the laboratory as soon as possible, preferably within 1 hour of the time you masturbate. Bring it in on the day assigned, even if you have not had 2 days abstinence. If for some reason you can't collect the sample on the day assigned, bring it in on the next day. When the semen sample is delivered to the lab, a few drops will be placed onto a microscope slide and videotaped through the microscope. The videotape is later played back through a computer system which will determine how many sperm are produced, how many are swimming, and how fast they are swimming. 2 other slides will be made and stained. These slides will be used to determine the actual shape of your sperm cells. Now, to review. Do not have sex for 2 days before you are supposed to collect the semen sample. Masturbate directly into the glass jar, fill out the information on the jar label, put the jar into the thermos, and turn it in as soon as possible. Thank you for your cooperation.